one and all. So let me introduce myself. I am Dr. Amar Ankalgi, working as professor and head in pharmaceutical analysis department. I am the innovation ambassador as per the IIC Institution Innovation Council. Today I am going to discuss about innovation in drug discovery and development, sophisticated instrument and its role in drug characterization. So as you all know that characterization of the drug is identification of a drug. So we generally synthesize new drug molecule, new drug moiety through various synthetic approach or through the natural product. Once we get that drug product, after that, that novel drug product structure must be characterized. To characterize the structure of a drug molecule, we either use spectroscopic technique and spectrometry technique. Okay, so that I am going to discuss today. So structural elucidation or characterization. There are different ways you can characterize the compound. Characterization is a part of the identification, and if we use a different instrumental tools and we combine it to finally confirm the structure of a compound, then that word is a characterization. For characterization of any compounds, we have a different techniques: mass spectrometry, NMR, IR and UV. Then you have a NMR, X-ray. These are the different techniques. For example, mass and NMR, it helps to find out the molecular formula of a compound. And regarding the functional group identification, the preliminary study is the infrared spectroscopy. For further confirmation of hydrogen and carbon situation in any compound, hybridization of carbon, we can go for the 30 carbon NMR or 1 proton NMR. UV, just you can find out the chromospheric group identification, functional group identification by the UV techniques. Beside that, we have a NMR, we have a X-ray. So X-ray is a technique where you will get a three-dimensional structure of a compound. X-ray stereography or X-ray diffraction techniques. Through that you can find out the structure of a compound. Whatever the DNA structure is there, that double helical strands of the DNA structure, it was confirmed by X-ray technique. Okay, so these are the different techniques you can use. You can get a 2D structure of a compound, and finally 3D structure you can, through virtual screening, you can confirm the structure. So these are some techniques that can be utilized for the characterization of a new drug molecule or existing drug. Also you can use these techniques. So for example, spectral interpretation is there. I have shown one spectra of the 1 proton NMR. So in 1 proton NMR, it is a one-dimensional spectra. It ranges from this is -2 to 12. We generally say 0 to 10. So it is expanded form. So here you can see we have observed the different peaks. Based on the peaks, we can characterize the protons present in a compound. Is it clear? So we have this CS2 multiplet we are getting here, CS3 multiplet you are getting, OH you are getting a singlet. Through that you can say that here compound is this propionic acid. This is the structure of propionic acid. The structure will be confirmed by this spectra, like singlet at 11.7, that is a OH, quadrate at 2.3, that is a CS2, triplet at 1.3, that is a CS3. So based on that you can confirm the structure by 1 proton NMR. Likewise, nowadays for easy performance we can combine different technology, like we can hyphenate the gas chromatography with the here mass spectrometry, GCMS. So whatever the injection you are injecting the compound, it is coming out. Through that column it is passing, and you will estimate the quantitative aspect of a compound. After that, the Eluent from the column, you will pass into the mass spectrometry. There will be the fragmentation, then you will get the mass spectra, and based on that you will characterize the compound. So nowadays lots of the instrument they have, we use a hyphenated approach for the characterization of a compound.